It is October of 1898, inside a stone cathedral in Turin, Italy, and an amateur photographer named Secondo Pia is locked in a small dark room with his hands trembling over a glass plate. Minutes earlier, he had stood before the most controversial cloth in human history, aimed his heavy camera at it under the harsh light of burning magnesium powder, and captured what he believed would be an ordinary photograph. Now, as the image slowly rises out of the chemical bath under dim red light, something happens that no one in that room expects. The negative plate, the reverse image that should look strange and unnatural, is instead staring back at him with a face so clear, so three-dimensional, so hauntingly real, that Pia nearly drops the glass into the developing tray. For a moment he cannot speak. What he is looking at should not exist. A medieval piece of cloth has just produced an image that behaves exactly like a modern photographic negative, more than 400 years before photography itself was invented. That single night in a Turin darkroom is the moment the Shroud of Turin stopped being a quiet religious relic and became one of the most fiercely investigated objects in the history of science. And more than a century later, a different kind of investigation, one buried inside strands of DNA too small to see with the naked eye, would push this mystery even further than Secondo Pia could have imagined. If you want to understand exactly what scientists found hidden in the genetic code of this cloth, and why it left some of them sitting in stunned silence, stay with me until the end, because this story does not end with easy answers. It ends with more questions than anyone expected. Let's start with what the Shroud of Turin actually is, because most people have heard the name without knowing the details. It is a long rectangular sheet of linen, roughly 14 feet in length and 3.5 feet wide, kept today inside a specially built case in the Cathedral of St. John the Baptist in Turin. Looked at under normal light, the cloth carries a faint brownish image of a man, front and back, head to head, as though the body had been laid on one half of the sheet and the other half folded over the top. The figure appears bearded, with wounds visible on the wrists and feet, puncture marks scattered across the scalp, consistent with something pressed into the head, long narrow marks across the back and shoulders consistent with repeated scourging, and a wound in the side of the chest. There is no detectable pigment, no brushstrokes, no dye worked into the fibers. Whatever produced the image did not sit on top of the linen the way paint does. It appears to have altered the very surface of the threads themselves, at a depth so shallow that it affects only the outermost layer of fibers, roughly a few hundred nanometers thick, thinner than a human hair sliced into hundreds of pieces. For more than 6 centuries, nobody has been able to fully explain how that happened. The documented history of the cloth begins around 1353, in the small French town of Lirey, in the possession of a knight named Geoffroy de Charny. From Lirey, it eventually passed through Chambéry, where in 1532 a fire broke out in the chapel holding it, scorching the folded linen and leaving burn marks and patches still visible today, before the cloth was moved to Turin in 1578, where it has remained ever since. Millions of pilgrims have filed past it during its rare public exhibitions, some describing the encounter as the most powerful spiritual moment of their lives. The Catholic Church itself has never made an official declaration that the shroud is authentic. It has also never dismissed it as a fraud. That caution exists for one simple reason. Science, despite more than 100 years of trying, has never been able to close the case either. After Pia's 1898 photograph, the shroud could no longer be treated as an ordinary curiosity. If it behaved like a photographic negative, then whoever or whatever produced the image had encoded something that the human eye could not properly decode without modern chemical technology that did not exist for the vast majority of the cloth's known history. In 1976, physicists using a device called a VP8 image analyzer, originally built to translate the brightness variations in satellite photographs into three-dimensional topography, fed in a photograph of the shroud almost as an experiment. What came out stunned them. Ordinary photographs and paintings collapse into a flat, distorted mess when run through that kind of analyzer, because the darkness of an image usually has nothing to do with the distance between the camera and the subject. The shroud did the opposite. It produced a coherent, anatomically correct, three-dimensional relief of a human body, as though the intensity of the image had been encoded based on how far each point of the body was from the cloth. A painting has never been shown to do this. In 1978, a team of more than 30 American scientists, physicists, chemists, and forensic specialists formed the Shroud of Turin research project known as STURP, and were granted 5 days of essentially unrestricted direct access to the cloth. They ran X-ray fluorescence, infrared spectroscopy, ultraviolet photography, and microscopic fiber analysis. Their final conclusion, published after years of review, stated plainly that they could not identify any pigment, dye, paint, or known artistic technique capable of producing the image, and that the mechanism behind its formation remained unexplained by the science available to them. That verdict has never been overturned. It has only become more complicated. STURP's investigation also went further than the image itself and looked closely at the wound pattern on the figure. Forensic-minded researchers going back to the French surgeon Pierre Barbet in the 1930s had already noted that the marks scattered across the back and shoulders appear consistent with a Roman flagrum, a multi-tailed whip fitted with small metal or bone weights, producing dumbbell-shaped bruising repeated dozens of times across the body in a pattern that matches known Roman scourging practice, rather than anything a medieval forger would have had reason to invent from imagination. The wrist wounds, rather than sitting in the palms as centuries of European religious paintings traditionally depicted, sit at the base of the hand, in the area anatomists call the space of Destad, a detail that later medical analysis suggested would actually be capable of supporting a hanging body's weight, unlike the palm, which would tear under that same load. A medieval artist working from religious paintings of the era, almost all of which showed palm wounds, would have had no obvious reason to invent that anatomical correction on his own. Now let's get to the part of this story that pulled the Shroud out of the realm of photography and physics and into the realm of genetics, because in 2015, a team led by geneticist Gianni Barcaccia at the University of Padua, working with colleagues including Alessandro Achilli at the University of Pavia, published a study that would eventually be seen as one of the most important scientific investigations of the cloth ever conducted. The raw material came from a very specific source. In 1978, during the STURP investigation, adhesive tape samples had been used to lift microscopic dust and fiber debris directly from the surface of the shroud, and small portions of that material had been carefully preserved for decades afterward by Professor Pier Luigi Baima Bologna, a forensic medicine specialist who had spent much of his career studying the cloth. Barcaccia's team was given access to this preserved material and set out to answer a question nobody had approached with this level of precision before. If they sequenced every fragment of genetic material clinging to the shroud's fibers, human, plant, animal, and microbial alike, what story would it tell? The technique they used is called metagenomics. Instead of hunting for one specific gene or one specific species, the researchers extracted and sequenced every recoverable strand of DNA in the sample simultaneously, then sorted the results by comparing them against known genetic databases. What came back was staggering in its diversity. Plant DNA on the shroud matched species from across the globe, from European spruce and Mediterranean clover and ryegrass, to rare East Asian pear and plum varieties, to plants found only in specific pockets of the Middle East. But it was the human mitochondrial DNA, the genetic material passed down almost unchanged from mother to child across generations, that produced the most striking numbers. According to the analysis, roughly 55% of the identifiable human genetic lineages traced back to Near Eastern populations. Close to 39% matched lineages, most commonly found on the Indian subcontinent. Less than 6% could be linked specifically to Western Europe, the exact region where the cloth is documented to have spent the last 600 to 700 years of its known history. Among the specific genetic markers identified was a mitochondrial haplogroup called H33, a lineage that shows up with unusual frequency among the Druze, a religious and ethnic community that has lived in relative isolation in the mountains of the Levant, in modern-day Israel, Lebanon, and Syria for centuries. Because that community has experienced comparatively little genetic mixing with outside populations, a Druze-linked marker is treated by geneticists as a fairly specific pointer toward that exact corner of the Middle East. Alongside it, researchers also detected lineages such as H2A2 and H1B, both more common across Europe and Central Asia, layered on top of everything else like sediment. Sit with that combination for a moment. A cloth with a fully documented paper trail running through 14th century France and 16th century Switzerland and Italy is carrying human genetic fingerprints that skew overwhelmingly toward the Near East and South Asia, with European genetic material barely registering by comparison. Barcaccia himself, when the results came in, said publicly that he found it difficult to believe that ordinary contact in medieval France, priests, nuns, pilgrims, and devotees moving through French and Italian chapels, could realistically account for such a strong genetic signature from the Indian subcontinent. His interpretation, stated cautiously in his own words, was that the linen itself may have originated in India, since the region historically produced some of the finest linen in the ancient world, and the Greek word for the shroud, sindan, has been linked by some historians and biblical scholars to Sind, an ancient textile-producing region along the Indus. If that reading is correct, the raw fabric could have entered the Mediterranean world through ancient trade networks that connected the Roman world to South Asia centuries before the shroud's documented European history even begins. But before you assume the case is closed, it is important to say clearly that not every scientist agrees with that interpretation, and a good storyteller has to tell you both sides. Geneticists writing in Scientific American after the study pointed out that the same tape samples also contained DNA from cats, dogs, cattle, pigs, carrots, wheat, corn, peanuts, and bananas, and that nobody is suggesting the shroud spent time in a Southeast Asian banana plantation. Historian Andrea Nicolotti, one of the more prominent voices skeptical of the shroud's authenticity, has argued that a piece of cloth publicly displayed, handled, restored, and touched by countless hands across 7 centuries in France, Switzerland, Belgium, and Italy would naturally accumulate an extraordinarily wide genetic mixture, simply because Europe itself, and especially its churches and pilgrimage routes, hosted travelers, traders, and worshippers from every part of the known world throughout the medieval and early modern period. In other words, a diverse genetic signature does not, by itself, prove where an object began. It only proves who has touched it. That distinction matters, and it is the central tension running through almost every scientific claim made about this cloth. It is also worth remembering just how much traffic the Mediterranean world carried in the centuries before and after the shroud's documented European history begins. Ancient trade corridors connected Roman ports to the Levant, to Egypt, to the Arabian Peninsula, and onward to India and beyond, moving silk, spices, dyed textiles, and travelers in both directions for well over 1,000 years before the shroud surfaces in 14th century France. Any object that passed through a handful of major trading or pilgrimage hubs along that network, even briefly, could plausibly pick up genetic hitchhikers from dozens of distinct populations without ever needing an exotic single dramatic explanation. That is precisely why serious researchers on both sides of this debate keep returning to the same conclusion, that DNA alone, without a fully documented chain of custody, can describe where an object has been touched far more reliably than it can prove where that object began. Now here is where the story takes a turn that even seasoned shroud researchers were not fully expecting. In March of 2026, the same research team, Barcaccia and Achille, working again with material collected during that original 1978 sampling, published a far more advanced follow-up study, this time using full metagenomic sequencing capable of reconstructing not just isolated fragments, but an entire reconstructed microbiome living on the cloth. The results confirmed and sharpened much of what the 2015 paper had suggested. The haplogroup H33, the marker associated with Druze populations of the Near East, showed up again. So did H2A2 and H1B. But the newer analysis went further, using kinship analysis to compare the DNA on 3 separate shroud samples against the genome of Baima Bologna himself, the very forensic scientist who had collected and preserved the material decades earlier. The results confirmed that a portion of the recovered DNA was a close match, or even an identical match, to Baima Bologna's own genetic profile. Direct proof that even careful, gloved scientific handling leaves a genetic fingerprint behind, and a sobering reminder of just how easily contamination can enter a sample handled across nearly half a century. And then came the detail that genuinely caught researchers off guard. Buried inside the reconstructed microbiome, alongside ordinary skin bacteria like Cutibacterium and Staphylococcus that you would expect on any object touched repeatedly by human hands, the team identified archaea, an entire separate domain of single-celled organisms distinct from bacteria that were specifically halophilic, meaning they require extremely salty, high-sodium conditions to survive. These are not organisms you find drifting through the air of a European cathedral or clinging to the walls of a storage vault in the Alps. Halophilic archaea thrive in environments like heavily salted, preserved foods, salt flats, and famously the Dead Sea, one of the saltiest bodies of water on the surface of the planet. Alongside them, the team found fungi including Dibariomyces hansenii, a species associated with cheese rinds and other extremely salty environments, and skin-associated fungi common to human contact. The presence of an entire community of organisms that can only survive in extreme salinity, embedded in the fibers of a linen cloth now kept in dry, climate-controlled Italy, strongly suggests that at some point in its history this shroud was stored, preserved, or kept in close proximity to a highly saline environment. The Dead Sea sits in the same region where the Near Eastern genetic markers, the Druze-linked haplogroup, and the earlier pollen findings all converge. 3 completely independent lines of biological evidence, human DNA, microbial DNA, and as we will get to, pollen, all quietly pointing toward the exact same corner of the ancient world. Because DNA is not the only biological trail investigators have followed on this cloth. Decades earlier, in 1973, a Swiss criminologist named Max Frey-Sulzer, the founder of Zurich's first forensic crime laboratory and the inventor of the tape lift method now used across the world to collect trace evidence, was invited to examine the shroud as part of an official investigating commission. Using his own tape lift technique, Frey-Sulzer spent 2 years analyzing pollen grains trapped in the linen fibers and reported finding species native to the Middle East, several of which, he claimed, grow only in the region stretching from Jerusalem toward the Jordan Valley and bloom specifically in early spring. If accurate, that would place actual plant pollen from that region physically inside the fibers of the cloth. But Frey-Sulzer's credibility took a devastating hit a decade later. In 1983, he was called on as a handwriting expert to help authenticate a set of documents that had just surfaced in Germany and were being sold to the press as the personal diaries of Adolf Hitler. Freisulzer put his professional reputation behind their authenticity. Within months, the diaries were exposed as an elaborate modern forgery, hastily written on paper containing a whitening agent that had not even been used in manufacturing until after the war ended, and one of the most embarrassing frauds in 20th century journalism collapsed in full public view, with Freisulzer's name attached to it. Critics of the Shroud have used that scandal ever since to cast doubt on his earlier pollen findings, arguing that if his judgment failed so completely on the diaries, his single, unrepeated pollen sample from the Shroud deserves the same scrutiny. Defenders of his work point out that his role in the Hitler Diaries affair concerned handwriting analysis, a completely different discipline from his doctoral training in botany and his decades of respected forensic pollen work, and that no one has ever successfully reproduced or independently retested his original tape samples to confirm or disprove them, because much of that material was never made fully available for follow-up study. What is left is an unresolved, uncomfortable footnote sitting right next to some of the Shroud's most tantalizing physical evidence, a reminder that even celebrated experts can be spectacularly wrong, and that a claim this important cannot rest on the word of one man and one unrepeated test. Then there is the radiocarbon dating, the test that many people still believe settled this question once and for all. In 1988, a small sample was cut from one corner of the Shroud and divided among three of the most respected radiocarbon laboratories in the world at Oxford, the University of Arizona, and the Swiss Federal Institute of Technology in Zurich. All three labs, working independently, returned strikingly consistent results, dating the linen to somewhere between the years 1260 and 1390, squarely inside the medieval period and matching almost exactly when the Shroud first appears in documented French history. For years afterward, this was treated as the final word. But the story did not end there. In 2005, chemist Raymond Rogers, who had personally worked on the original 1978 STURP investigation and had access to the actual threads removed during the 1988 sampling process, published a chemical analysis arguing that the specific corner used for radiocarbon dating showed clear evidence of having been rewoven at some point in its history, a common medieval repair technique used to disguise damage along an edge, blending newer thread invisibly into older fabric. Rogers based this on differences in the chemical composition of dye and cotton fibers, found only in that corner and absent from the rest of the Shroud, along with vanillin loss measurements, a natural chemical marker that decreases at a predictable rate in ancient linen over time, which suggested the main body of the cloth was chemically consistent with being significantly older than the radiocarbon corner alone would indicate. If Rogers is correct, the 1988 test may have accidentally dated a medieval patch, rather than the original fabric, which would mean the true age of the cloth is still an open question rather than a closed one. It is important to be honest here. Rogers' conclusions remain genuinely contested within the scientific community, and plenty of qualified chemists still consider the original 1988 radiocarbon date to be sound. This is not a case where one side has been definitively proven wrong. It is a case where two carefully conducted scientific investigations reach conclusions that are difficult to fully reconcile with each other, and that tension has never been resolved. There is also the matter of blood. Long before the DNA studies, Bima Bologna himself, examining the reddish-brown stains positioned exactly where the wounds on the figure appear, identified them in the 1980s as consistent with human blood of the AB blood type, a comparatively uncommon type globally. That finding has been debated for decades, with some researchers questioning whether centuries of aging, environmental exposure, and prior handling could reliably preserve a testable blood type at all, and whether pigments like iron oxide could mimic some of the same chemical signatures under early testing methods. Like nearly everything else connected to this cloth, the blood typing claim sits in that same uneasy middle ground, taken seriously by some specialists and treated with real skepticism by others. So where does all of this actually leave us? A photograph developed in a Turin darkroom in 1898 revealed that the image on this cloth behaves like a photographic negative centuries before photography existed, a property that STURP's team of more than 30 scientists in 1978 could not explain using any known artistic technique. A three-dimensional analysis in 1976 showed the image encodes distance information the way no painting ever has. Genetic testing in 2015 and again in 2026 uncovered human DNA weighted heavily toward the Near East and South Asia, specific Druze-linked genetic markers pointing toward the mountains of the Levant, and an entire hidden microbiome of salt-loving organisms that should not exist on a piece of linen stored for centuries in the dry cathedrals of Alpine Europe, unless that cloth spent meaningful time somewhere far saltier. Pollen evidence from 1973, gathered by a man whose later reputation would be shattered by one of history's most notorious forgeries, still points in that same geographic direction, unconfirmed, but never fully dismissed either. Radiocarbon dating in 1988 said medieval, plainly and clearly, and a 2005 chemical reanalysis raised a genuine, still unresolved question about whether that specific sample even represented the whole cloth. None of these findings, taken alone or even taken together, prove that the Shroud of Turin wrapped the body its supporters believe it did. That was never something science was built to prove. But after more than 125 years of some of the most advanced forensic, chemical, and now genetic technology on Earth being aimed directly at this single piece of linen, nobody, not the skeptics, not the believers, and not the scientists standing in the middle, has been able to make this mystery go away. Every time a new tool is invented, it gets pointed at this cloth, and every time it comes back with an answer that is stranger and more complicated than the one before it. The DNA did not close the case in 2015. The follow-up study did not close it in 2026 either. If anything, both investigations opened it wider, adding new layers of evidence that point toward the ancient Near East, while simultaneously reminding everyone involved just how easily centuries of human contact can blur the line between where an object came from and everywhere it has been since. Secondo Pia walked out of that dark room in 1898, convinced he had witnessed something science could not yet explain. More than a century of the best laboratories in the world have since confirmed at least one part of what he felt that night. This cloth still refuses to give up its secret quietly. Every laboratory that has taken its turn with this linen, from Pia's dark room in 1898 to the genomics labs of Padua and Pavia more than a century later, has walked away having learned something real, and every single one of them has walked away with new questions they did not have going in. That pattern alone, repeating decade after decade, technology after technology, is part of what keeps this cloth sitting at the center of one of the longest-running scientific investigations of any single object on Earth. If you made it this far, you now understand this story with more precision and more honesty than most of the documentaries built around it, because the real story was never as simple as one side being right and the other being wrong. It is a genuine, still unfolding scientific investigation, layered with brilliant insight, honest disagreement, and at least one damaged reputation along the way. Subscribe to this channel and stay close, because the next investigation into the mystery science is still trying to explain is coming soon, and you will not want to miss it.